I divided my talk in two parts, so I'll be quite quick. So if you don't understand much, you can ask questions. So one is about creating tools. One to try to elucidate gene function, and the other is to try to elucidate the mode of action of small molecules. So the first part of my talk is centered on this area of systems biology, which is called reverse engineering. And the idea is that you can use multiple perturbation to cell lines and collecting measurements of the state of the cell, which in our case for now is gene expression profiles, because these are quite easy and abundant to obtain and also to collect from databases, and then try to learn from these gene expression profiles a model of gene regulation, although you will see it's not really gene regulation, but it gives you an idea of what to look for. So the reverse engineering approach that we have used this time is quite simple and is very close to the correlation coefficient that maybe you have heard many times. Actually, I will use this one. And basically, the idea is this one, that if you have 3 genes and you would like to understand whether these genes have something to do with each other, where there's this something you will have to define later doing proper experiments. So this just gives you some hypothesis generation. Then what you can do is to collect a lot of these gene expression profiles following perturbation data. And what you have here in this figure is you have the level of expression of one gene, and you plot it against the level of expression of another gene, and then each of these points represent the change in expression of one gene in one experiment compared to the change of expression of the other gene in the same experiment. So if you have 100 different gene expression profiles obtained from 100 different experiments, you will have 100 different points over here. And then just by looking at this graph, this plot, you can say whether the 2 genes are related to each other or not. On the left side here, you see an example of when 2 genes are not related to each other. On the right panel, you see an example of 2 genes that instead are related to each other, because when one gene is increasing its expression in some experiments, the other gene is decreasing it. Whereas when one gene is decreasing its expression, the other gene is increasing it. So you can do this kind of, you can measure this relationship using a mathematical quantity that is called Motile Information. And we did that on a large scale using collecting all the available microarrays up to now, because I've been working in the last 7 years mainly with microarrays, which are now going away. So it's my goodbye to microarrays. So what we try to do here is to collect as many microarrays as possible from public databases, and we collected about 20,000 microarrays in human and 10,000 microarrays in mouse. And then using this Motile Information approach, what I just described before, we came out with a gene network where for each pair of genes, we compute this Motile Information. That is, we say whether they are related to each other or not. And if they are related to each other, we draw a line between the 2 genes. So we have what we call a network, a graph, which tells us about this association between genes. So just to give you an idea, we use the microarrays for human that contain 20,000 transcripts, and we used 20,000 different microarrays in human, and we came out with 5 million of these lines, of this association among these 20,000 transcripts. And we did a similar work in mouse, where we used the 10,000 gene expression profile covering 45,000 different transcript probes. So this, to give you an idea of what comes out, if you plot the top 1,000 strongest interaction in human, this is what you get. You see the biggest cluster of genes is ribosomal genes, that they are all co-regulated, so they all go up and down together in these 20,000 different experiments. And then you get other genes involved with the immune system and a group of genes involved with the cell cycle. Many of these relationship between genes are related to real protein-protein interaction. So many times these genes are changing together their expression because they interact at the protein level, and so probably is to maintain stoichiometry in the protein complex. And we did an experiment in which we took this set of genes here that is involved in a cell cycle. For some of these genes, it was known that they were interacting at the protein level, but we predicted many other interaction, and we assumed that this interaction could be protein-protein interaction. So these genes are co-regulated at the expression level. Maybe they are interacting at the protein level. And then we did some histohybrid experiments, and we confirmed that about 50% of these co-regulation or co-expression that we measure from microarrays are protein-protein interaction, at least at the histohybrid level. However, the limitation of these approaches is they don't tell you what these genes have to do with each other. They could also be completely indirect relationship. But I will show you that they can be useful for generating hypotheses on a gene of interest. And the way we use this network for this is to get a gene and to look at what are the neighbors of this gene in the network. By neighbors, we mean all the genes that are co-regulated, according to our analysis, with your gene of interest. So it's like you extract one gene from your network, you look at the genes with which it is connected, and then you can extrapolate the function of this gene with a classic approach that is called guilty by association. So you say, I don't know the function of these genes, but the majority of the neighbors of this gene, here I divided them in two groups, they're all neighbors of these genes, have to do something with the cell cycle. So I predict that my gene maybe is involved in cell cycle. So we did this selecting 16,000 genes for which a biological function was known, at least from databases. We assumed that we did not know the function, and we tried to predict the function of this gene by looking at the neighbors. And depending on what we were trying to predict, we were correct 60 to 70% of the times. So this is just to say that this network can provide you with some hypothesis on the function of your gene, because you can look at the genes with which your gene of interest is co-regulated and make some hypothesis. To prove that this was the case, we looked at genes that we predicted to be involved in lysosomal function. How you do that? For each of our 20,000 probes in our network, we looked at the neighbors and we predicted the function of the gene. And we put this in a database, and now you can go in the database and ask for a biological function of interest or a cellular localization, and you can get out the predictions on all the genes that we made. So, for example, here we asked whether there were genes that were predicted to be involved in the lysosome function. Why lysosome? Because it's a very hot topic in my institute currently, so I chose this one. And we saw that these are the prediction confidence that we have. And the first genes, the one with the highest confidence, was this Niemann-Pick disease gene, which is a known lysosomal gene. And the second gene with a very high confidence was granulin, which is a gene that causes neural degeneration, dementia. It is not known to have any function related to lysosome. It is known to have a growth-like activity, and its function is not clear at all. But we had this prediction that it has, it is involved in lysosome. And so we did some experiments quite easy. One was to, in collaboration with Nicola Brunetti at TIGEM, one was to treat cell with sucrose which are known to increase lysosome levels. And we saw that this is cathepsin D is increasing, and also our granulin gene is increasing. Then we overexpress the TIGEB, which is a transcription factor that has been shown to regulate many of the lysosomal enzymes. And by overexpressing TIGEB, here is the overexpression of TIGEB, we see that the level of granulin is increasing a lot. We also looked for binding site for TIGEB in the granulin promoter, and we found the binding site. And here instead is a LAMP2 immunofluorescence assay, which shows that when we treat cells, we overexpress granulin in the cells, we have an increase in LAMP2 fluorescence. And when we get the medium of cells that are expressing granulin, and we use the medium on wild-type cells, again we see an increase in LAMP2. We also did the electron microscopy, I don't show it here, where we see that we have an enlargement, not an increase in the number of lysosomes, but an enlargement in the number of lysosomes. Now we are going forward with this story, not myself, because I don't care about lysosome actually, but what this was to show is that a prediction that is very easy to do with our tool drives you some hypotheses that then you have to follow up if you're interested. I'll go now quite quickly. This is just to show you a view of the human network at the genome scale. We ordered here genes according to their chromosomal position, and you have a black point if two genes are interacting. That is, they are co-expressed according to our analysis. And we verified that genes belonging to the same chromosome have a higher tendency to be co-expressed. And this is very clear in chromosome 19. Maybe you can see that it is a little bit darker than the rest. However, you can reorder the genes, not according to their chromosomal location, but according to if they have the same neighbor, so if they are co-regulated with the same genes. And if you do that, you can identify very clear modules within this network, and we could show that these modules are indeed enriched for biological functions. Another thing that we did was to then check whether genes that are co-regulated have something to have a link with the chromatin structure within the nucleus. In order to do that, we used recent data that were developed in which using high-C chromatin capture, you can do at the chromosome level or at the genome-wide level, you can check whether two regions of the chromatin with the resolution of 1 megabases are in contact in the nucleus. So where you see red here, it means that these two different regions of chromosome 14 have a higher probability of being close at the chromatin level in the nucleus. And here what you see is the contact probability for chromosome 19. This is the centromere region. And as I said, if you see a red spot over here, it means that this 1 megabase of chromosome 19 is in contact with this 1 megabase of chromosome 19 in the nucleus. So it's like a spaghetti, and there are these parts of the spaghetti that are close to each other. And here instead you see the co-regulation of genes belonging to these 1 megabases. So you can see that genes that are close to each other at the chromatin level tend to be co-regulated in our network. And this is very striking. I mean, here you can see it, and the p-value is very, very high, is to the minus 100, because you can see that things that are close at the chromatin level are also co-regulated. Okay. So this is the conclusion of my first part, and you can access all our network at this website. It's called netview.tigem.it. This work is in publication, but the database is free. You can just go in, put your email, and then automatically you will receive a password. And you can put your gene of interest and see what we predict, what are the neighbors, or you can select a biological function of interest and see what are the genes that we predict belong to that function. Now, going to the drug network, which is not a Neapolitan drug thing, but is something to predict the mode of action of a molecule. The work before was from a PhD student of mine, Vincenzo Belcastro, graduated, and now he's at Philip Morris. And Francesco also is a former PhD student, is now at EBI. And again, we created a tool. This time, this is called mantra.tigem.it. And what the idea of this tool is to try to identify the molecular pathway that are targeted by a small molecule. So if you have a new small molecule, you can use this tool to identify what this small molecule can be doing, or also for drug repositioning. So if there is an already known drug, we wanted to see whether we could reposition it to have another mechanism of action that was not known before. Another thing that we are now doing, and also in collaboration with Stefan, is using this drug network to predict what could be the possible drugs that could be helpful in treatment of diseases. And I will give you an example later. So here again, we are using microarrays. We are waiting for RNA-seq data now because microarrays are not popular anymore. But now microarrays were very useful because this was this large data set that was done at the Broad Institute at MIT, where they treated 5 different cell lines with over 1,000 small molecules. So for each molecule, they treated 5 different cell lines at different dosages, and they measured gene expression profiles via microarray in a treated cell versus controlled cells. So for each small molecule, you have several gene expression profiles depending on the cell lines and on the dosage that tells you what are the genes that are upregulated or downregulated following the drug treatment. So what we did was first to merge all the expression profile for one drug, because the problem in this data set is that you have a small molecule of interest, but you have many different gene expression profile, and if you look at the genes that are upregulated or downregulated, they are different depending on which cell lines you're looking or which dosage you're looking. So we first wanted to create a synthetic gene expression profile by merging together all the gene expression profile obtained with the same drug on the different cell lines and the different dosages. We used an algorithm that is similar to Google page ranking, what Google does is to get the choice of each of us when we do a search and then merge them together to give you a list which is more close to what that is a consensus of what everybody was looking for. And this is the same. We created a consensus list for each small molecule. So for 1,000 drugs, now we have 1,000 list of genes that are up or down regulated, the consensus list. And then we generated the measure of similarity between the transcriptional profiles of 2 different drugs. So for each drug, we have multiple expression profile. We created a synthetic gene expression profile, which has a consensus list of genes that are up and down regulated. And now for each pair of drugs, we look at how similar the 2 synthetic transcriptional profiles are, and we generated the distance between 2 drugs. So this number, this distance is small if the 2 transcriptional profiles elicited by the drugs are very similar. And then we can plot these into a network where each point here now is a drug, and we have to a line connecting the 2 drugs if these are very similar according to our distance measure. That is, they elicit a similar transcriptional response in their profile. And what we discovered is that when you look at this network, and now is you find groups of drugs that are very closely interacting with each other. So they are very similar to each other. So this is a group of drug here, but they are very different from the other drugs. And we identified 106 different groups of drugs. We call these drug communities. So this is completely automatic, just based on transcriptional profiles. But what we discovered is that if you look, for example, at this group of drugs, we called it the group 100, this is all antipsychotic drugs. And they are very similar to each other according to these transcriptional profiles, although they are done on cell lines that probably are not depressed or have problems. But there is a very strong transcriptional. And again, we have cardiac glycosides also have a very similar transcriptional response, and they are very different from others. Then we have HSP90 inhibitors, HDAC inhibitors, and so on. So we saw that we could automatically group drugs that were very similar with the mode of action. Then we asked, can we use this network to predict the mode of action of a molecule? So if now you have a new molecule, and we did this in collaboration with Antonella Isacchi from Nerviano Medical Sciences, so they were developing anti-tumorogenic compounds, and so what they did was to treat different cell lines with their compounds. They sent us the gene expression profiles following treatment versus control, and it was completely blind. They did not tell us what their drugs were. This was a real blind test, Napoli, Milano, so it was very... so they didn't trust us. And so we then told them what their drugs were, and they were very surprised because we could, for example, in this case, there were HSP90 inhibitors that we grouped together in this group 28 in yellow, and the 3 drugs that they sent us were HSP90 inhibitors, and we could tell them, look, these are HSP90 inhibitors. And we did this for 9 different compounds, among which CDK inhibitors and topoisomerase inhibitors. So we could prove, and now you can do it yourself on our web tool, that if you treat your cells with the drug, and then you compute the gene expression profiles, you do not have to do any statistics. So you do not have to tell us which are the genes that are significant. You just have to put on the website the list of 22,000 22,000 probes in human. Is a self. No, you have to process the self file, but you just have to tell us to rank the genes between the ones that are overexpressed and the ones that are downregulated, and you give the whole list without doing any statistics. And then we place your drug in this network, and we tell you what we think your drug is doing. Another way that we tried to use this drug was to reposition a small molecule, as I told you before. And in particular, again, because it's a very hot topic in my lab because of our director interest, is autophagy. And so with Nicola Brunetti, however, not with Andrea Balab as the director, but with Nicola, we decided to see whether we could identify molecules that are already in clinics, because this set of 1,300 molecules is FDA-approved drugs that could enhance autophagy. In order to do that, I asked Nicola to choose one molecule between these 1,000 that was known to increase autophagy, and he chose 2D-deoxyglucose. And then we looked at our network, which were the small molecules that were they were closer to the deoxyglucose, so that elicited, according to our analysis, a transcriptional response that was very similar to the deoxyglucose. And these are the 4 most, the closest drugs to do deoxyglucose, and 3 some are antipsychotic and so on, but they were known to increase autophagy, and one that is called Fasudil, over here, which is used in Japan for over 20 years as a vasodilator. We predicted that Fasudil could enhance autophagy. To prove this, you already know now about LC3, 1, and 2 very well from the previous talk. So what we saw was treating with Fasudil is number 2 compared to untreated cell. We saw an increase in LC3, 2, and we used as comparison also rapamycin as a positive control, and other and 2-deoxyglucose is number 4, and this other drug, which is an antipsychotic, but it is known to induce autophagy. And also by immunofluorescence, we could prove that fasudolin did increase autophagy. Then this is a preliminary work that we have done that we have not continued because Francesco has finished his PhD, and so now we have to continue, but we are collaborating with Stefano. And however, the idea is this one. Instead of taking a profile of genes that are differentially expressed because of treatment with the drug, can we take a disease and look at the genes that are differentially expressed in disease versus control, and then put it in the drug network and predict what could be the drugs that have either a similar transcriptional profile or an anti-similar transcriptional profile, so they could probably restore the balance. So it was so to do that, the first thing that we did was to look in the literature for microarrays that were done on in this case we found Parkinson. So these are post-mortem brains of Parkinson patients, and they were done, now I don't remember, it was like 6 patients and 6 controls. We generated the prototype, this consensus list of differentially expressed genes, so we had a single list. And then we looked at which were the drugs that were more similar and which were the drugs that were more anti-similar. So the drug that was most similar to this Parkinson was isoquinoline, I think is pronounced, which we found is used in mouse models to induce Parkinson-like symptoms. And instead, the more anti-similar was this sulindac sulfide, which is a dopamine level reducer. So this was quite interesting because using a disease transcriptional signature, you can correlate it to small molecule and either better understand the pathogenesis or maybe make hypotheses on possible small molecules that could be useful. And also we did the collaboration with Stefano, but you will read it probably soon, the results. And again, we starting from data from patients, we could select drugs that were similar or anti-similar to the transcriptional profile. And this is basically the work of these 2 people in my lab. That they are now not there anymore, but the tools are still there on our website, so you are free to use them. So thanks a lot for your attention.